human genome, the first 3 billion string of letters that encode the DNA in a human, was published in 2001. It took 11 years and about $3 billion. All of the grant dollars, all of the work that was done, it was estimated that to get one full human gene sequence was about $3 billion, and it took about 11 years. Okay? So let's do everybody's sequence. Yeah, we got a bit of a problem. Okay. But in 2017, the technology has gotten so good that it has moved so rapidly that what took 10 years now takes about a day to do a whole genome sequence. What cost $3 billion in the 90s to get it all done, now in 2017, a whole genome can be done for under $1,000. And I fully expect by 2020, we will have a $200 genome, a standard clinical cost test that will give the sequence of an entire genome. That is a rapid drop in pricing and the advances that have been made because of the technology. And the technology is referred to as massive parallel sequencing that I won't get into. But we now have the capability to do what took 11 years, now in one day. What took $3 billion now costs about $1,000. In a few years, it'll cost $200. So let's focus on the technology. And instead of saying, what should we be looking at, we now can consider all of those questions. We can ask the questions about the genetic diversity, the genetic heterogeneity, and if we can do it in a cost-effective way, and we can do it rapidly, we can do it on a lot of people. So there's the technology that's developing right now. What have we learned already from doing those kind of sequences? Well, we know there are a lot of changes in the DNA sequence. Part of the problem is if you sequence a myeloma cell, you're going to say, well, you know, it's got differences in the DNA sequence. They're not present in the normal cells of that individual. And part of what we're trying to figure out, and this is the term people are using, what genetic variations in the myeloma cell are drivers, and what are passengers? And what passengers I mean, there's an inherent genetic instability, and some of the changes in the DNA in myeloma may have nothing to do with malignancies. They're just being carried along. Now we have a problem saying, well, how do we figure out which are the drivers, which are the passengers? We have to sequence lots of these and find out what are the common drivers and what's the noise of the backstreet driver, okay, the passenger. Some DNA changes may be associated with risk assessment, high risk versus low risk. Some DNA changes are seen at relapse. So look at the sequence of the myeloma at diagnosis, it's treated. When it relapses, what changed? And if we understand what changed, we have a better idea of what to do about it. Okay? So part of the problem is myeloma, with the technology that we now have available, we learned one thing. It's a complicated disease. There are a lot of genetic events happening in this. And part of the, you'll hear on Sunday, is the COMPASS study. You heard about Black Swan and the idea of this minimal residual disease. You'll hear from Joan Levy on Sunday, the COMPASS study, and this is the Multiple Myeloma Research Foundation, and they're looking at sequencing these 1,700 target genes, asking the question, what's different in those genes in different patients? What's different in those gene sequences in a relapse versus a diagnostic sample? And all of that starts giving us better ideas of the biology of the disease. And then I'll also say something on Sunday about we can also use genetics to start asking questions about toxicities. Who's prone to a toxic event? Who's likely to get a neuropathy? Who's not likely to get a neuropathy? It's based on your genetics, and we're learning more about the causes of that. So of course, we have a lot more data to analyze, and you can't read this probably, but the doctor's now saying to the patient, your medical problems are more complicated than I thought. I'm going to refer you to another doctor who has more medical insurance than I do. Okay? Which is kind of being passed on to somebody that knows a little more, and they can charge you more. Okay? All right. But the DNA sequence is only one measure of variability. The other thing is, remember that DNA is made into this messenger that makes the protein? Well, that's the expression of the DNA. It's expressed into these messenger molecules. And different people have different levels of expression of the genes that are in their genome. So somebody might have a lot of an oncogene and somebody has a little bit, and it can make a big difference. So the amount of expression of the genes in that code can be very important. And I'll talk more about that on Sunday because it gives us a hint about response, resistance, and toxicity. So on Sunday, I'll talk more about gene expression, toxicities, looking at one cell at a time, what we can learn from that, using computers to sort through all this, because it is complicated, and then a little bit about the Mayo University collaboration that's happening. So let's go back to the DNA problem. So there's genetic variation, there's mutations, there's rearrangements, there are all these things happening in the myeloma. So what if we could change the DNA? What if we could edit it, change it? Okay, and this is now gene editing. And it was the winner of Science Magazine's Method of the Year. The Method of the Year in 2011 was called gene editing. That is modifying DNA sequence to change function. That is, introduce a mutation, if you find a mutation in a myeloma and you go, is that a passenger or a driver? One way to find out is to isolate some cells, genetically engineer those cells to have the mutation and ask, did it create a disease by changing that sequence? Or fix a mutation. If you've got a bad gene, make it a good gene. So, or knock out a bad gene, or knock in a good gene. These are all genetic manipulations that people are doing, and there are technologies to do that. When you do that, it requires certain technologies. One of those is what we call a vector. You have to somehow get that DNA sequence you want to change into the cell. And there's one organism that does a really good job of getting DNA into cells: viruses. So what people have done in research labs is they've commandeered a virus and they change the virus DNA to a DNA of choice. And then when the virus attacks the cell, it shoots the DNA into the cell. Normally the virus does that to create more virus, but they've hijacked the viral component of injecting DNA into cells and are now using that as an approach to get DNA in to change sequences in cells. But you got to make sure you hit the right target. You got to change the right gene. You got to be pretty efficient at it. I mean, if you do it 1 out of 1,000,000 cells, that's great. You may have changed one cell, but you got all the rest of them that you didn't do anything. It has to be sustaining, and it can't create off-target problems. As soon as I start introducing DNA into cells, it can go places I don't want it to go. It can affect other genes that I don't want it to affect. And I'll show you in a minute. That could be a real problem. So the challenges are, what gene do you want to fix? Do you want to knock it out? Do you want to edit it? Is one gene enough? Should we fix just one? Or should we fix 6? We got to figure that out. How many cells do you have to affect? Well, presumably all of them, right? So that's a challenge. Will this approach work in cancer? Are there safety concerns? Are there ethical concerns? We're changing DNA. We're changing life code. And there are a lot of people that have problems with changing life code, saying, you kind of blame God here in altering DNA sequence. So the question is, should it be regulated? So the challenges have been, number one, vectors like viruses were pretty good at injecting DNA, but the DNA didn't go to the places it was supposed to go. So it was an inefficient delivery. It was really bad targeting. It created serious problems. It often didn't sustain. And then the really bad thing is that there were a number of cases where genes were putting into kids to correct an immune deficiency. And there was a famous trial in France where about 14 kids a gene was inserted into their genome to correct a severe immunodeficiency. Good news is it worked. The bad news is half the kids came down with leukemia because the DNA was going into places and activating genes that cause malignancy. And so in 2003, as the New York Times reported, the Food and Drug Administration said, stop. You can't do this anymore. It's not safe. So they halted 27 different gene therapy trials because the danger was now a complication of the gene therapy. The good news was we got the DNA in, we corrected the disease, but we created more problems. So that stopped. And now the whole promise of editing the genome became a real problem. People said, we failed. We failed to deliver on the promise of gene therapy. Along comes a new technology. This is the CRISPR-Cas technology. I'll guarantee you, in a few years, this will be a Nobel Prize. This will be a Nobel Prize. CRISPR stands for Clustered Regular Interspace Short Palindromic Regions. Name is not important. What it is what, again, just like we hijack viruses to inject DNA, what researchers found was that simple organisms like bacteria, they're prone to being infected by other bacteria or viruses that infect the bacteria. And they can avoid that by using a system that changes the DNA sequence of the invading DNA. It's like bacterial immune system, okay? And it was first discovered in E. coli. Those are the bacteria in the stools that you produce. That's your gut bacteria, E. coli. And then one of the systems that they discovered also, this Cas9, comes from Streptococcus, which is the bacteria that causes strep throat. But that wasn't the issue. They pulled out just certain pieces of those bacteria that perform these functions. And the scientists cleverly adapted this to do everything I said was important to do. Getting the DNA in, doing it efficiently, targeting it appropriately, not having off-target effects. And Jennifer Doudna and Emmanuel Charpentier at Berkeley kind of pioneered this method. And then Feng Zhang at the Broad Institute in Massachusetts really took it and started applying it to all types of things. And now there's a huge patent fight. They both have tried to patent this technology. So there's a huge patent fight going on with that. The idea is, this is DNA surgery. Surgery using this system called CRISPR. CRISPR is the DNA vehicle. And what it can do is it can precisely locate the gene you want to correct. It doesn't go places it's not supposed to go. It uses this guide system that they discovered to correctly target. And it works in any animal. It's highly efficient. It's very specific. It doesn't have this off-target effect. It's quick. You literally can order this system as a kit online. This system comes with component parts that you can order all the pieces. And as long as you have a sequence that you know you want to target, you can order it through company, okay? So you can knock out a gene, you can knock in a gene, you can fix a gene, you can even fix 5 or 10 genes at once with this system. This is Nobel Prize winning stuff. So you can buy your own gene editing kit, and if you want to go to that website, they'll sell you a kit, okay? So, first human clinical trial using CRISPR. This was a Google search, okay? The proposed trial was developed by Dr. Carl June at the University of Pennsylvania. And what he wanted to do is he wanted to figure out a way to amp up the immune system so that it would attack myeloma cells, would attack cancer cells. So they wanted to reprogram these cells here called T cells, and these T cells can target other cells and kill them as part of your immune system. And the plan was that all the scientists will recruit 18 patients with 3 types of cancer. One of those cancers is myeloma. So the first human clinical trial that's using the technology of CRISPR to modify T cells to attack tumor cells, one of the 3 cancers chosen was myeloma. And that is being done at Penn, and this is Joe Biden with Carl June, and then the myeloma expert at Penn is Ed Stadtmeyer at Penn. So they're involved in this trial. It's a very low number of patients. They want to see if they can effectively genetically engineer T cells to attack myeloma cells. Not any T cell, the T cell of the patient to attack their own tumor, okay? The other thing that people have been doing, as I mentioned, is this whole idea of an avatar. Now there's an interesting title, Intratibial Injection of Human Myeloma Cells into NOD-STID IL-2R gamma null mice mimics human myeloma and serves as a valuable tool for anti-cancer strategies. Don't make me say that more than once. That's a mouthful. But the bottom line is these mice can be injected with human myeloma cells. They fluoresce. You can see them as they fluoresce. And you can try different treatments and ask which treatment effectively eradicates the tumor from the mouse, which combination eradicates it from the mouse, and therefore you have a laboratory test for your individual myeloma can be put into that mouse, and 17 different drug combinations can be tried to ask which is the best one. Okay, that's in development. And then there are the immune therapies, the new things like daratumumab and elotuzumab. These are the new immune therapies. Basically, in these, what they're doing is they're generating an antibody that will recognize your tumor cells, and they do it because they recognize these molecules that are on the surface of tumor cells. Name's not important, one's CD38, the other one's called SLAM7. We know, based on lots of people analyzing it, that looking at different proteins that coat the surface of myeloma cells, they contain CD38 and SLAM7. Not a lot of other cells contain those. So now if you can raise an antibody in a mouse, in a pig, in a sheep, in a goat, if you can raise an antibody by injecting that CD38 protein into the goat will mount immune response. You isolate the antibodies and give the antibody back to the patient. The antibody will recognize the CD38, and the other side of the antibody will link up with one of these natural killer cells. Natural killer cells, when they get activated by an antibody, nail the nearest cell nearby, which will be a myeloma cell. They literally will secrete products that will completely lyse that cell and break it apart. So this is a case where both of these antibodies are being used to target the surface of a myeloma cell and recruit your own natural killer cells to mount an immune response of your immune system against the myeloma. Interestingly enough, some of the early trials, they weren't all that dramatically effective. So the daratumumab and elotuzumab, as single agents, they had some effect, but people weren't being put into complete remissions by adding these antibodies. But when they added them to Velcade and IMiDs, the added effect was very dramatic. So there are actually some very exciting new clinical trials that are being developed where part of the combination therapy now is going to be something like a Velcade or carfilzomib plus dexamethasone plus an IMiD plus one of these immune cells. The important part of this is it's using your system to kill tumor cells, and hopefully that starts reducing some of the toxicity. I'm not going to go. So has myeloma changed in the past 10 years? Well, myeloma itself has not changed in the past 10 years all that much. It's still complicated, but as the doctor says here, good news, your cholesterol has stayed the same, but the research findings have changed. Okay? So, well, okay, what does that mean? The research findings are doing a better job of understanding and defining the individual nature of everybody's tumor and where the vulnerabilities are, so that when you walk into a doctor's office, the goal of the future is not to say, I'm going to treat you along with 700 other people with the same drug. I'm going to look at your myeloma, I'm going to characterize it, and I'm going to build the cocktail of drugs that works best for your particular myeloma. And what about the cost of new therapies? Well, as it says here, the trial meds are free, but the bottle costs $3,000 a month. Okay? So what do we gain with all this new technology? We gain specificity. We gain individualized approaches to target your myeloma cells. They become more effective, less toxic, they can be sustained, and the goal is to get it down below the minimal residual disease by using these new types of technologies. Thank you. All right, you are.
Ladies and gentlemen, Bob Tuft has got a brief comment to come and give us.
My comments are brief, partly because I'll be throwing things at you. Originally, Dave said you could bring a fungo bat and hit balls to people in the crowd. Have my wife Suzanne, my caregiver, who I love as my who's also an attorney, she thought it was probably not a good idea. And considering the hour and our age, advancing age, I'm in the young old group too. Thought it was different. So, Don Baylor and Bob Tufts autographed baseballs. I'm going to give 2 questions, 2 baseballs. We have more tomorrow. Tomorrow's questions will be baseball and cancer related. First one's easy. Name the 3 oldest ballparks in the major leagues. People get 2 out of 3.
Don't know what he's talking about.
Wrigley Field.
Wrigley
round
Field is number 2.
of cricket.
Park's number 1.
Wrigley.
Wrigley and Fenway. Everyone gets those.
LA.
Pardon?
LA.
Which one?
Dodger.
Dodger Stadium is right.
Wow, good job.
I was going to throw it. We'll break on it. See what happens. Number 2, second one. Sandy Koufax didn't pitch first game of the 1965 World Series because it was Yom Kippur. He pitched games 2, 5, and 7. Pitched game 7 on 2 days rest, to a 4-0 win with only giving up 4 hits. Which pitcher that should be in the Hall of Fame pitched for the Twins that day? Who was known as a broadcaster and an excellent fielder, an all-around damn good person. No Minnesota people? Come on. .287 career wins. Pitched mostly for the Twins. Also got a hit. What? Earlier. 1965. This guy pitched in parts of 4 decades, from 1959 through the early 80s. I thought there was an older group. Come on.
No baseball fans.
Or I'll have to give her the alternate.
Bob Feller.
Then, which pitcher who went to Stanford was the Cy Young Award winner for the Boston Red Sox in 1967? Retired and became a dentist afterwards.
I'm on. You don't want to throw it at me. Throw it at him.
And we're done.